Right, so I'm going to talk for about 25 minutes on this topic, and then some of my colleagues are going to speak each for a few minutes each. So I'm going to talk about high-throughput sequencing in trauma infections, put this rather fanciful subtitle towards Microbiology 2.0. Going to talk about high-throughput sequencing, applications in clinical microbiology, some local case studies. In fact, the promise of benchtop sequencers, Nick Lohmann's going to talk more about that, what we propose to do in this, and perhaps where future microbiology will be. Just put one little point up though as a starting point, which Edison said about genius, 1% inspiration, 99% perspiration. I think a lot of what we're doing is driven by instrumentation, and we're trying to stay unashamedly trying to stay at the cutting edge of the instrumentation, always trying to get the latest instrument and use it, and that underpins much of what we're doing. So if we look at what's used to be the case, what is still the case in some areas, we have sequencing invented in the 1970s with Sanger dideoxy sequencing primarily, genome sequencing coming in the 1990s. Both approaches rather time-consuming, expensive, low throughput, and really not suitable for the average laboratory, either in a hospital or in a university environment, and dedicated, and certainly at genome level, stuck in institutes. Epidemiological typing, lots of different methods out there. And even where you have the same kind of method, like MLST, it has to be customized for each particular organism. Different labs do it in different ways and so forth. The point is, all of this is time-consuming, it's onerous, and it's often underpowered as well because many isolates get bundled together as indistinguishable. And so you might say you've got a load of isolates on a unit that are all indistinguishable. You can't work out who's been giving it to what. Under those conditions. So high-throughput sequencing erupted on the scene about 5 years ago with the promise of doing sequencing 100 times faster, 100 times cheaper than conventional sequencing. In fact, we're probably now 1,000 times faster, 1,000 times cheaper. In a sense, it's what you might call a disruptive technology. It's a kind of game changer. And there have been a number of different technologies come into the marketplace already. Very lively competition between them, and most of them, if not all of them, are showing year-on-year progress in terms of throughput, ease of use, cheapness, and so on. These rely on fundamentally new approaches, different from conventional genome sequencing. So instead of propagating things in E. coli on plasmids, you actually kind of grow molecular colonies, and you have new approaches to actually reading the sequences in those colonies as well. So kind of one of the issues I'd just like to flag is that, if we look at diagnostic microbiology these days, in many ways, certainly in bacteriology, I mean virologists perhaps are a bit more future gazing, but in bacteriology a lot of what we do is harking back to the time of Koch and Pasteur, looking down microscopes, growing colonies on solid media. And perhaps a future vision is that we can just do everything through sequencing. That's kind of just a naive, hand-waving kind of vision, but it's something to test, to push against, to fight against, and use as a way of seeing how far we can take that vision. And I have written a little piece in Genome Medicine recently pointing out that it is naive to think that we could ever do everything by sequencing, but let's see how far we can go. So this is a slide from Nick Lohmann, just pointing out, once you've got a genome, there's all sorts of things that you can do with bacterial genomes. You can identify organisms, detect them, diagnosis, typing, epidemiology, population biology. You can reconstruct, although you're looking at the genotype, you can reconstruct the phenotype and the metabolic processes going on within the organism. In one case, famous case, that people were able to devise a culture medium for an organism that was previously unculturable by looking at the genome. You can look at pathogen biology. You can look at the evolution of the organism, how it becomes resistant to antibiotics, how, if it's a chronic infection, how that organism is adapting to life within the host. You know, in cystic fibrosis, that's one particular theme that people are very interested in. When we're looking at sequencing methodologies, though, we're not just looking at genomes, we're looking at these two independent approaches, two different approaches. We can look at culture-dependent approaches where we actually still have to grow stuff, and then we pick it off the plate and do things. And there we can genome sequence the purified DNA and get a whole genome sequence out at one end, at the other end. Or we can look at culture-independent approaches where we don't actually have to put anything on agar plates. And there are really two kinds of ways of doing that. One is what we might call molecular barcoding. A particular molecule called 16S is very popular for that, but there are others out there. Or you can just sequence all of the DNA in the same way as you sequence the DNA when you're looking at a colony. Here you just sequence all the DNA in that community, and that we call metagenomics. The first one we call phylogenetic profiling or community profiling. So over the last few years, a couple of years now, there have been a number of papers showing proof of principle in this area. As I say, virologists are always the first off the block, and there's been a number of examples where they've done high-throughput sequencing in a completely unbiased way, just taken cDNA from patients and from tissues where it was unclear what was going on, clear that the patients were sick and that there was an infection but didn't know what the cause was, and identified new viral causes and new kinds of virus that had not been described before. And in several of these studies, they actually went from having the genome, from doing the metagenomics or metatranscriptomics, getting out part of the genome of the virus and then devising specific, much easier to use diagnostic reagents that they could use for it in a kind of simple PCR and so forth. For bacteria, things have lagged behind a bit. There are a few papers out there until recently that people were doing it, but not in anger very much. This group here in Texas, probably leading the field there. One paper there, early paper of theirs, looking at the polymicrobial nature of chronic diabetic foot ulcer biofilm infections. And I came up with this rather annoying acronym there, BTFAB, but generally what they're doing is using 454 to sequence the amplicons, 16S amplicons from these samples to see what lives in there. Just in the last few weeks, so there's been the same group have been doing more extensive studies and studies that actually are closer to what we intend to do. Here they're looking at venous insufficiency wounds using conventional culture approaches and molecular diagnostic approaches. And I just grabbed this from PubMed this morning. The paper itself is still in the proof state. It's not actually as a marked up proof yet. It's just what's been submitted to the journal, but you can download it. And so what they did was they compared 3 different systems, and I think Captain Kirkup actually mentioned this study this morning. Basically, they showed that if you look, you can see a lot more using the high-throughput sequencing approaches. You get many more species and genera, bacterial genera out there, and that you can identify wounds that were not going to heal very well from the community profile that you're getting out of the sequencing that you're doing. And here's another paper that they've had in recent months, where they've shown that if you look at wounds and then you look at what's in your microbiological profile using these approaches, and you tailor your treatment based on that, you take into account what you're finding from the molecular methods rather than just looking at what you see in culture, and tailor your treatment that way, you actually end up with a better outcome. In terms of genomic epidemiology, these are the kind of papers that got me interested in this sort of area. Back in the 90s, it was shown that a dentist, on the balance of probability, well, beyond reasonable doubt, actually gave HIV to his patient because they obtained sequences from the dentist and the patient, and showed that they were too close for it to be just by chance. The Amerithrax incident stimulated a lot of work in this area, looking for polymorphisms in Bacillus anthracis. That was about 7 or so years ago. And in the last couple of years, we've seen a crop of papers, several crops of papers, in fact, now come out, looking at this, looking at various organisms, Streptococcus pyogenes, MRSA, tuberculosis, M. tuberculosis. In fact, another study, actually a definitive study on the Amerithrax incident, actually nailing down the culprit, the person that did it, and the seed lot that he used to prepare what he put into those envelopes. I grabbed this particular article because I was talking in the southern United States a few months ago. In this, they sequenced the leprosy bacillus genome from around the world, and particularly focused around America. And one of those facts for a pub quiz: the only other animal that gets leprosy apart from humans is the 9-banded armadillo. And it turns out that wild 9-banded armadillos in the US have leprosy. And these guys showed that, and there were some strange people in the US who go and capture wild animals and skin them and eat them. And these guys showed that actually there was transmission from the armadillos to humans. They also showed in the bigger picture that the armadillo leprosy originated in humans in the first place. So we gave it to the armadillos, and then they are occasionally giving it back to us. So, watch what you eat when you go to New Orleans. The political ramifications of all this kind of stuff are illustrated by this paper in the New England Journal of Medicine, where they showed that the Haitian cholera outbreak through genome sequencing was very likely to have originated from the Nepalese UN peacekeepers that were in Haiti. And it created a big trouble in Haiti, as you might imagine, that this wasn't something from local, it was these people coming in and bringing it in. In Birmingham, we've had a 454 sequencer now for a couple of years. Nick Loman is going to talk after me, and myself, we both blog. Here's his blog entry. You can see how geeky we are. We are really our boffins. Nick would have been in Bletchley Park if we were in the Second World War. He would be working here. But here we are. He's working. He's saying that he's like a kid looking forward to Christmas. And here's my colleague Charles Penn standing, looking a bit like an idiot actually, next to the crate that brings the sequencer to us. So we actually, put pictures of the sequencer arriving on our blog. That's so much into it. But getting serious again, we've looked at a number of questions and seeing if we can address them in our pilot work that we've been doing, the kind of work we've been working up to more recently. So asking questions like, can we detect differences between isolates within an outbreak? Isolates that would otherwise be indistinguishable. Can we actually tell anything from those differences? Can we look at how resistance emerges? And general question, what is a species within a genus in microbiology? And we focused on one particular organism, Acinetobacter baumannii, which is a gram-negative bacillus. It's multi-drug resistant typically, and two agents, colistin and tigecycline, are kind of held as reserve agents. But this organism is probably one of the most, the furthest along in the progression towards being untreatable with any agents, so-called pan resistance. It's associated with wound infections, ventilator-associated pneumonia, sometimes bloodstream infections, and a particular issue has been that this organism, infections with this organism, have been associated with returning personnel from Iraq and Afghanistan, both in the UK and in North America, in the US as well. And there has been there have been case reports of transmission from military to civilian patients in that setting. And in fact, there was, a few years ago, it's calmed down a bit, there was an awful lot of anxiety about this. There were patient advocacy groups and all sorts. I think there still is a www.acinetobacter.org site detailing all the kind of woes. Problems with this, it's hard to identify in the clinical laboratory. There are these so-called genome species that are very, very similar but not quite the same, but you can't tell them apart very easily. You can do various kind of typing assays on outbreak strains, but we can't look into the modes of spread and transmission. And it's very hard to work out how it becomes resistant in individual cases. And we don't really understand how this works as a pathogen. So there was an outbreak here in 2008, and all isolates within that outbreak were all indistinguishable by current typing methods. We've written this up in the Journal of Hospital Infection. We used 454 genome sequencing of 6 isolates. We then looked for what we call single nucleotide polymorphism, single base changes in the genome that differentiated those isolates from one another. We were very careful not to be picking up any false positives. I mean, if you're looking at differences of 1 in a million between genomes, you have to be very careful that you're not just picking up sequencing errors. And so actually we did go through and validate anything that we saw using conventional Sanger sequencing of PCR amplicons as well. What we found was this. So we got 4 patients. The ones with M are military patients, the C ones are civilian patients. We found 3 loci in the genome where they differed, and these are the patterns of differences we saw there. Now when you look at that, you kind of think, well, what does that mean? The top there, the AB0057, is actually a completely unrelated isolate, which is had been genome sequenced, and we use that to try and work out the polarity of change. So we take that as the ancestral state for this organism, and then we can see various changes there. It makes much more sense, though, if we map that on to the time and space aspects of the outbreak. So there was some patients in a 6-bedded unit in the intensive care unit. Some of them were in the main unit, and one was in a trauma unit, completely separate, had nothing to do with any other patients while within the hospital. Now, if you look at that, you can see that there are 3 patients that have exactly the same genotype there, this TAG genotype, M2, C2, and M4. Now, it turns out that M2 and C2 were next to each other in adjacent beds for a period of time, and M2 had it before C2. So I think on the basis of parsimony, we can make a reasonable judgment that actually M2 probably was the source of the infection in C2. Actually, although these M2 and M4 didn't actually come across each other in a hospital, when digging back into the records, they actually were repatriated at the same time through the same pathways, and so it's likely that either one gave it to the other during that repatriation, or they caught it from the same common source along the way. So there was an epidemiological link there. Now you may be saying, well, isn't this an awful lot of effort? It's a sledgehammer to crack a nut to just get this bit of information, when you could have probably guessed that had happened anyway. Point was that this is our kind of benchmarking exercise. We didn't know at the outset whether there would be any differences between the isolates, or whether they'd be interpretable in any way at all. So we were generally quietly cautious after this. Another study we've done with collaborators in London is to look at 2 Acinetobacter baumannii isolates from the same patient, one of which was isolated before tigecycline therapy and was susceptible. The other one was resistant and came after therapy. And in comparing those, we actually found a larger number of SNPs, actually fairly large number, 18 of them. And one of those SNPs was in a known resistance gene, or at least in a gene that regulates a resistance phenotype within that organism. So that kind of that was nice and made sense and kind of validated the approach. An interesting thing that we didn't anticipate was that the later strain, the later isolate, actually had some deletions in it. Normally we think about resistance coming as a result of new DNA coming in, or of single base changes and so forth. But what happened here was that there were 3 deletions in the later isolate, and one of them actually truncated a gene called MUT-S, which is involved in DNA repair. It might be a just so story to say this, but we rationalise that to mean that probably what happened was that once that DNA repair was lost, the mutation rate went up, and that's why this strain actually then acquired a mutation that gave it resistance. We're also doing some work on looking at the genus Acinetobacter. We've genome sequenced 13 isolates from 9 species, and we've done various kinds of phylogeny and so forth to try and look at, can we get a consistent phylogenetic picture, a consistent taxonomy, and we're kind of playing with the idea, can you have bacterial taxonomy that's based entirely on genome sequences and ignores or any difficult things that you might like to do, like phenotypic characterization, hybridizations, and so forth. And most of the trees actually do give us a congruent picture. Interesting point, though, is that this 16S, which is often used as a barcode, seems to be the one exception, which is actually giving us a slightly different topology in our tree than other housekeeping genes like RPOB. I'll leave that out, I think. Another more recent development and all that was that back in February, I suddenly discovered I'd won a new instrument. So we had the 454 for a couple of years. Then Ion Torrent, a company based in the US, announced that they were bringing out a new instrument that was cheaper, quicker than the 454. And I chanced my hand and thought, oh, well, I might as well fill in the form. They wanted to have a competition in Europe to introduce it to Europe. They said they give away 2 instruments to people in this competition. So out of 150 entries, I was one of the 2 lucky ones that got the instrument. So we've been working with this and gearing up to use the technology. Not using our own instrument, but using the technology and then working with others in terms of analysing data coming off the instrument. We actually managed to get ourselves a New England Journal paper over the summer. It's us collaborating with German and American colleagues on this. This was the German E. coli that you probably heard about in the news. We were one of 4 groups to actually get this genome sequence, but we used Ion Torrent data, and also, interestingly, we engaged in what we call crowdsourcing. So we got lots of people from around the world to contribute to that, working with the Germans and with the Chinese in the BGI. If you're interested in that, you can read the paper, or if you just Google me, you'll find a talk I gave last week in Germany on that. So, finishing up now, what are we actually proposing to do within this proposal, within this centre proposal? And here's a list of the things that we said we would do in the proposal. So, first thing was to trace the spread of microorganisms, so do genomic epidemiology. And this is a back of envelope calculation. We estimated we could perhaps do 200 isolates a year. It may be that we can do more than that. As I say, the technology is getting better all the time, cheaper and so forth. It won't be 200 isolates of the same thing. We were going to have to, we've given ourselves enough space to come and look at different issues at different times. So if we have an Acinetobacter outbreak of 20 isolates, we might do those, and if we have an MRSA outbreak of 20 isolates, we might do those, and so on. We also want to do culture-independent diagnosis, the sort of thing that we've mentioned earlier and that Captain Kirkup mentioned. Again, we put down a ballpark figure of 100 samples per year. That, again, is a slightly movable feast. But this will give us a chance to actually look in detail at what's going on in these samples and see how that might impact on our understanding and even on the treatment of these patients. Several people have mentioned the fact that fungal infections are a problem in trauma patients, and unfortunately most microbiologists still rely on very old-fashioned approaches to identifying fungi. So we are aiming to exploit a number of different molecular barcodes, ribosomal RNA genes, mitochondrial genes, genomes and so forth, to try and develop novel, rapid, sensitive molecular approaches for this, particularly focusing on unusual filamentous fungi, which are a problem here. The numbering's gone wrong, but this should say 4 and 5. So, key point in doing any of this is we have to collect all the clinical metadata. So we've said that that's what we're going to have to do. We're going to have enhanced surveillance and better clinical data collection. There was a research nurse, or several research nurses written into the proposal, and we'll be able to draw on that resource to actually beef up the collection of data and make sure we do that. And then we have 2 PhD studentships, one that's going to go to the Hawkey lab and one to the Piddock-Weber lab, looking at this data once it's started to be collected and see what can be made from it in terms of understanding infections caused by staphylococci, extended spectrum beta-lactamase producing organisms, possibilities of seeing what happens when you use bacteriophage before and after on wounds using molecular approaches, and looking at resistance mechanisms in wound isolates. So, in closing, in the 19th century we had agar, or at least we had solid media, and we had microscopes. Towards the end of the 20th century, people started using DNA approaches, but I think in the 21st century we're going to start moving towards whole genome sequencing and sequencing as a kind of assay, as a diagnostic modality. And there's a certain kind of aptness in this, because DNA itself was first isolated from surgical pus, and so I say perhaps we should close that loop and say let's go back to basics and start looking at the DNA that's in pus by sequencing it routinely as our diagnostic assay. So that's just an acknowledgement slide, and